Okay, we're also recording, right? Yeah, so I think you can go this way. Yeah, all right. Let's see. Where are we? No, I'm missing the. Yeah, I'll make you join in a few minutes. Yeah. So I just want to kind of say that, we have been doing this auction for 20 years. And as you can see, this is how much of annual research funding we have in 2005 to 2023. So this was the COVID blip here. And we have funded more than $7 million in research funding, and we are really proud about that. And we are 100% volunteer organization. And we want to really thank all of the researchers who have been with us for all these years. And later on in the auction, you will see a slideshow of how young people were to 20 years later. So if you see any of those people around, please say hi to them, all the researchers and scientists. And thank you for all the presenters today. And we are going to start with our first presenter. So Jeff has just arrived. So we are going to start with Stephen Tapscott, and then we'll do Jeff.
Any seats taken?
So now we'll play this. And you should be able to do that.
Okay, thank you, Premi. And it's great to be able to talk to you today and tell you about some of the work we've been doing on FSHD, work that the patient community and the FSHD community has really been instrumental in, and the work that Friends of FSHD has been very instrumental in. This is a schematic of early human development. You get the egg fertilization, and then that divides 2 or more times to get to the 4-cell stage. And up until the 4-cell stage, the early embryo is living on its maternal RNAs and maternal proteins. And then it's only at that 4-cell stage, the embryo starts making its own gene products and activating its own genome. It's called zygotic gene activation or embryonic gene activation. And if you think back to those early times of your life, you might remember the first thing you did was turn on DUX4. DUX4 is a transcription factor, and it activates that first wave of embryonic gene activation and really starts the embryo developing. DUX4 is a transcription factor, and it activates many genes that have many functions during embryogenesis. It regulates the expression of genes like ZSCAN4 that maintain the telomeres, the parts of the chromosome necessary for constant replication. Other transcription factors like EOMs that regulate the development of the placenta and the mesenchymal cells in the embryo. It rewires what's called the epigenome, the way the chromatin is packaged to make genes accessible or inaccessible through several different mechanisms. And it induces many other genes, including retroviruses and repetitive elements in the genome. So it really has a profound effect on kicking off the beginning of the program that leads to in the end, a formed human being. DUX4 then turns off, and it's supposed to be off for the rest of your life. But in FSHD, because of a genetic change that weakens the lock of keeping it off, you get leaky expression of this very powerful transcription factor in skeletal muscle. And it activates the same program in skeletal muscle that it was evolved to activate in the early embryo. And I won't go through the details of the consequences of this, but you can imagine that activating an early embryonic program in skeletal muscle is really detrimental to muscle function. Not only that, at that early phase, at the 4-cell stage, when DUX4 turns on and you transition from the maternal RNAs or the maternal proteins and maternal program to the embryonic program, that's associated with shutting off that early program so you can turn on the embryonic program. And we and others have recently shown that in addition to turning on the embryonic program when DUX is misexpressed in skeletal muscle, it also shuts off the myogenic program in the skeletal muscle. And so you can see that that would lead to a lot of difficulties in skeletal muscle function. So as I was saying, when the DUX4 is misexpressed in skeletal muscle, it does the same thing it does in the early embryo. It activates that early embryonic program, induces many proteins that are normally only expressed in the early embryo, and your immune system has not been tolerized to, and so it could be inducing expression of proteins that lead to an immune response. And there are many other molecular factors that DUX4 does that can lead to disease. Dennis Shaw, Seth Friedman, and others have shown that when you do MRI scans of skeletal muscle in an FSHD individual, and these are two different images, T1 and STIR of a lower extremity from an individual with FSHD, and Seth will be talking about this in greater detail later, you see some abnormalities on the imaging for MRI in the FSHD muscle. T1 really measures fat content, and T2 measures free water, which is often associated with edema or inflammation of the muscle. Normally, muscles have a very dark characteristic on both STIR and T1 signaling. Early in the FSHD phase, there's an inflammation or accumulation of free water, and that creates an early STIR signal. And then that eventually leads to replacement of the muscle with a fatty infiltration, and you convert from the STIR signal to the fatty infiltration signal. Some years ago, when the Seattle Wellstone started including FSHD in its research program, again, really with the help of Friends of FSH Research, we wanted to find out what the underlying pathologic and molecular correlates were of those MRI changes. And so some of you in this room may have participated in the study where we enrolled 36 individuals in the study, did MRI of the lower legs, and then biopsied muscles with particular MRI characteristics to try to determine what those underlying molecular changes were and histopathologic changes in the muscle that correlated with the MRI. And this resulted in 2 publications really showing that those early inflammatory changes were associated with what we thought, inflammation, but also the increase in DUX4 expression and the expression of genes and proteins induced by DUX4, which may have been underlying the initiation of that inflammatory process. We could then do RNA sequencing, where we look at the expression of all the RNAs in the genome, and this is a heat map where each row across is a different gene, and each column is a different muscle biopsy. And you can see if you compare control muscles to what we call which were the STIR positive muscles, you get this hotter expression, which means higher RNA expression of the genes that are regulated by DUX4. And you get other baskets of genes activated too, genes associated with inflammation, extracellular matrix, and so the consequences of the DUX4 expression. But then you also, in the muscles that have not progressed to STIR positivity, that are relatively MRI normal, you also have the hints of these genes, same baskets of genes being expressed compared to the control. And so this underlying low level of activity that then progresses to higher levels of expression as the muscle becomes STIR positive. And that led to a good model for disease progression that integrated the MRI findings with the molecular findings of an initial mild phase with the low level of DUX4 and target gene expression, to then sort of increasing ramping up of that expression that then was associated with the MRI positivity, and then later on fat infiltration. More recently, we followed that up with essentially a validation study where, again, some of you may have participated in this. We enrolled 34 individuals and did MRI functional testing and muscle biopsies of the anterior tibialis muscle, both the left and the right muscle. The results of this study were just this last week published in Human Molecular Genetics, and I'll just go over some of the results from that study. This is a plot of the data where you have the DUX4 score on the x-axis going up, and then a measure by MRI of whole muscle fat content on the y-axis going across. The level of the DUX4, and this is DUX4 score, which is the basket of genes regulated by DUX4. The higher on the axis, the more expression of DUX4. If you look at controls, they're really lower than any of the FSHD subject biopsies, the expression of in the muscle of the FSHD subject biopsies. And then as the DUX4 score gets higher, you transition from the blue dots, which were that STIR negative MRI, to the red dots, which are the STIR positive MRI. And if you conclude the percentage of fat in the muscle based on the MRI characteristics, you can see at about the 0.2 or 20% fat level, there's this transition to this MRI STIR positive and very high DUX4 levels. And so this is going to be helpful to us because when we do clinical studies to test whether a drug improves FSHD, we can choose muscles that we have a high probability of knowing will have a high level of DUX4 expression. And so, for example, on the right here in the STIR positive muscles, if you look at the average expression of the DUX4 score, it's higher than in the control muscles. And even in the STIR minus muscles with that lower level of DUX4, those are still higher than the control muscles. And if you look at the other baskets, you can see that we can now molecularly characterize the effect of the DUX4 expression using the molecular markers for muscles that we've chosen based on MRI characteristics. And obviously, the goal of an intervention is to identify a muscle that has the correct MRI characteristics for fat infiltration, STIR characteristics. So when we do a biopsy, we know we're going to find a muscle that is involved with FSHD pathology and gene expression, intervene with a drug and show that we can decrease that amount of DUX4 score or the other basket scores down to that intermediate level of mild affected early disease characteristics, or hopefully down to the control level. And so this will be a measure of whether a drug is active and successful earlier on in a clinical trial that will give us an idea whether the drug is working and worthwhile pursuing to make it into a full phase 3 trial for functional activity. Just one other thing that we learned from this study, which was really, I think, important for us to think about and plan for future studies, is that there is really a strong correlation between the scores in the baskets, like the DUX4 score in the right anterior tib, as well as in the left anterior tib. And this shows if you had a perfect correlation where the right and the left were really the same, it would be a perfect 45-degree line going through there. And you can see in the upper left-hand panel, the DUX4 score is really pretty good right to left. But what is best is this panel down here, which is the immunoglobulin score, which is genes expressed by B cells. And we have other evidence that T cell genes may have a similar characteristic. And this has really brought us to a new awareness to look at circulating immune cells as a potential mediator of some of the progression in FSHD and giving us a new venue to look at. We don't know how productive this venue will be, but it's something that's come out of the study that is a new opportunity for us to think about alternative approaches to therapeutics, in addition to some of the many other great approaches you'll hear about today. And so we did get our Weston Center renewed recently, and I'll just tell you the first aim of the new center going forward. Some of you may hear from us about this. It is an extension of the initial patient cohorts we had to continue to do MRI and functional studies so that we can then go back to the early molecular studies we did and say, what are the predictors of disease progression in the muscles that we biopsied some years ago? There's no repeat biopsy in this study. And then we'll also be doing some of the more sophisticated MRI analysis that Seth Friedman will be talking about later. And then the second part of this phase of this new phase in the Weston program is to do safety study with clenbuterol, a drug that we had identified years ago that is able to suppress DUX4 expression in skeletal muscle. And it's a dose escalation study for safety, but then we'll also be using it as a training study to use the MRI and the molecular outcomes that we've developed to demonstrate that they are effective in monitoring response to therapy in this context. And I just want to thank the many people who were involved in this study. I won't go through the list, but I also want to just call out Xiaojian Wang, who will be at the meeting tonight, who's the bioinformatics specialist who really was instrumental in doing this. I want to thank you for coming, and Friends of FSH Research for really being instrumental in getting these projects off the ground.
Are there any questions for Dr. Tapscott? We can get like one, maybe two of these.
I have a question regarding the biopsies of the TAs, because they're so, depending on the fatty content of what you're extrapolating. Wasn't Joel Chamberlain doing something around non-invasive biopsies, and is there any on that?
Do you read the other questions?
The question was looking at some of the changes in the muscles. Is there an approach to non-invasive biopsies to look at that? Well, there's the MRI that Seth will be talking about later. People have been doing impedance myography and ultrasound, but I'd rather let other people speak to those approaches.
How soon do you think you'll be able to see a drop in those basket scores that you presented during the clinical trials?
Yeah, I think right. It's speculative at this point, but if we just pick, how quickly we'll be able to see the response of the basket scores following an intervention in clinical trials if the intervention is successful. The DUX4 target score should go down nearly immediately because we would be turning off DUX4. They're very short-lived factors. That should go down very quickly. Some of the other factors, such as extracellular membrane, might take weeks or months to normalize, but we have rational models for each of the categories and how quickly they would revert.
Hi, yes. Did I hear you say, so going STIR positive, I presume that could be a one-way trip, but you seem to indicate that you could go back to STIR negative?
That's a really good question. The question was, if the treatment is successful, will you revert from STIR positive to STIR negative? That's a really good question. If it's simply due to inflammation and an active inflammatory process, we should be able to shut that down if the treatment is successful, and then you would revert back to STIR negative. However, we actually don't know the real fundamental architecture that underlies that STIR positivity, and so that's something that we'll have to answer as we go along.
So STIR negative doesn't imply that you have increased function, just reduced inflation. It could still be a damaged muscle, but it's just not inflamed. Is that right?
Yeah, so the follow-up question was that STIR negative doesn't mean the muscle is normally functioning. And what I can answer in that is, in the natural progression of the disease, the STIR negative muscles are generally normally functioning. And then even as they go through the early STIR positive phases, they're only beginning to lose function at that point. The intervention, whether that would revert you to a functioning muscle or just stop progression, is an open question.
Right, that's all the questions that I'm sure Dr. Tapscott would be happy to answer.
Hey there. Right at the beginning. So, yeah, so I just wanted to give a brief introduction to our center. Stephen told you a little bit about it. I'll go over a few other things though. But let me tell you a little bit about gene therapy, because the project, one part of our center and most of the work in my laboratory is focused on trying to develop gene therapy for different types of muscular dystrophy. So this is just a general overview of the history of gene therapy. It was actually first proposed in the scientific literature way back in 1972. And that was at an early stage when I don't think anybody had a clue how one could possibly do human gene therapy. And as an example of that, the very first patient that was enrolled in a human clinical trial for gene therapy was back in 1990. And the field was progressing reasonably well for a number of years. And then it hit a major roadblock in 1999 due to the death of Jesse Gelsinger in a clinical trial for gene therapy of a liver disorder. And that was a major setback for the field, as you can imagine. The people had a lot of reservations about gene therapy, you know, whether it was going to be safe and effective and things of that nature. And then to have a patient die created all sorts of issues, both politically, scientifically. It became very difficult to get funding to continue working on gene therapy. And in some ways it was, well, in many ways, I thought it was rather ironic, because if you think about the history of advances in medicine, one example is the development of organ transplantation, in particular heart transplantation. And the first several hundred patients that received a heart transplant died within about a month of that transplant. And yet a single death in the gene therapy field completely shut down the field for a couple of years. Fortunately, it kept going again. It took a while to ramp things up, find ways to make it a little safer. And I think the first successes we started to see in a variety of clinical trials was in about 2009. And then it wasn't until 2017 that the first gene-modified cell products became approved for use in the United States by the Food and Drug Administration. And this was the development of these so-called CAR T cells. I don't know if anybody has heard of those. Those are becoming increasingly used for a variety of cancers, particularly hematopoietic cancers like lymphoma or leukemia. But the first gene therapy for more of a solid organ was approved by the FDA in 2017. It's called Luxturna, and it's a treatment for a rare inherited form of blindness. And it's partially effective, but it restores partial vision to the patients that have this disease. A huge breakthrough came 2 years later in 2019 with the approval of Zolgensma, which was one of the first really systemically delivered gene therapies. Zolgensma is a drug for spinal muscular atrophy. It falls into the category of neuromuscular disorders. And the way Zolgensma works is you have a gene therapy vehicle that's introduced right into the bloodstream, and it goes all over the body. And by the way, I should point out that this discovery that you could deliver genes body-wide by introducing a gene transfer vector directly into the bloodstream was something that was discovered in our Weston Center here in Seattle, and it's now being very widely applied throughout the world. Last year, there was a breakthrough in that the first gene therapy for a muscular dystrophy was approved by the FDA. That's known as Elevidys. It's the fifth gene therapy so far approved in the United States, but the first one, as I mentioned, for an actual muscular dystrophy. And I can also point out that the basis for this drug was also developed in our Wellstone Center here in Seattle. Just last month, or maybe 2 months ago now, the FDA approved the first gene therapy that was using a form of gene editing. Some of you maybe have heard about gene editing. CRISPR-Cas9 is mentioned a lot in the lay press. A form of that was approved for treating sickle cell disease, which is one of the most common genetic diseases in the world. And the FDA came out last month and announced that they think they're going to approve up to 17 more gene therapies in 2024. So we're really well on our way towards the advancement and the application of gene therapy. So how does that relate to our Wellstone Center? Well, one of the goals of our center has always been to try to develop gene therapy. And as you probably heard a little bit from Stephen's talk, we kind of have two parts to our center. One is more basic development, trying to understand the basic biology of some of these muscular dystrophies. And then there's a focus on translating those discoveries into clinical action. And a major way to do that is through the development of gene therapies. And as I mentioned, we've been successful so far with at least one form of muscular dystrophy. The other aspect of our center is to really focus on understanding the disease better and doing clinical studies. So the history of our Wellstone Center here a little bit is that, again, it was established almost 20 years ago now. Originally, we were only studying DMD because that was the muscular dystrophy we knew the most about. And some of the key discoveries that have been made over the years were major breakthroughs in understanding how to switch genes on and off to make so that they'll make their protein products in the appropriate tissues. We had to find ways to make genes a lot smaller than they normally were so that we could manipulate them in the laboratory and try to deliver those back into patients. I mentioned we discovered the first and really the only method that's out there now on how genes can be delivered body-wide, and that was this intravascular method using the so-called AAV vectors. That was a key discovery in our center. We've identified a lot of safety challenges for gene therapy in large animal models. And as I mentioned, our studies led to the first gene therapy for muscular dystrophy that was just approved last year by the FDA. So our Wellstone Center is supported primarily by the NIH, but we receive a lot of help from the Friends group supplementing our grants. Typically, these centers are supported for 4 to 5 years at a time, and then you have to reapply to NIH and put in a new proposal. There are 6 Wellstone centers throughout the United States. Our center was the first one that was funded, and it's the longest running center currently. And we, as Stephen mentioned, we were just renewed last September for another 5 years. So we're very excited about that. And as you've heard, it's a collaborative approach with a number of groups throughout the U.S. And our focus has shifted really after the very first grant from Duchenne to an even split between studying Duchenne muscular dystrophy and FSHD. So the organization of our center has 2 main projects. One is to try to identify and overcome some of the key limitations to developing gene therapy for muscular dystrophies. Our focus here is heavily on, again, Duchenne and also FSHD. And Project 2, as Stephen told you about, is focused more on the clinical foundations of FSHD and is supported by 3 different core laboratories to oversight developing gene therapy vectors and also a training mission that we have to supply fellowships and interactions between scientists and clinicians. And of course, we have a very close relationship with the Friends of FSH. So a little bit about Project 1, which is focused on gene therapy, as I mentioned. So again, the history, we developed really still the only known method for body-wide gene delivery to muscle that's being used in clinical trials all over the world right now. We developed synthetic miniaturized genes for DMD gene therapy, translated those discoveries out of the laboratory into biotechnology, and this led to the FDA approval that I mentioned earlier. What we're working on now is to really try to adapt those methods to other muscular dystrophies and as well as heart failure. But the focus of the Wellstone Center itself is to try to advance the current DMD gene therapy vectors that were approved. They're only partially effective, and we have some ideas on how to make them far more effective. And then we're trying to adapt those to FSHD. And the way that that's being done, the gene therapy for FSHD is primarily being led by Joelle Chamberlain at the UW. The idea is to use the same backbone that's being used in a lot of the other muscular dystrophy gene therapy trials, which is an AAV delivery shuttle. And in contrast to a lot of gene therapies where you're trying to deliver a new gene to replace a defective or a mutant gene, the idea with FSHD is to try to deliver a genetic cassette that will actually destroy the mutant DUX4 transcript. So it's a sort of the opposite of what you do with more typical gene therapies. And the testing that's being done for that involves muscle cells in petri dishes, a lot of development in mouse models, and then new collaborative work with Project 2 and with Peter Jones at the University of Kansas to try to adapt this to a new pig model for muscular dystrophy. So actually, I'm just going to stop there then. So if there's any questions, if we have time, I'd be happy to answer to you.
Probably have time for one question.
Yeah, you were just saying that you had ideas on how to make the AAV vectors more effective for DMD. One of the little different is that applicable to FSHD.
Can you repeat the question?
Yeah, fine.
Yeah, you can.
Talking into the microphone here. Yeah, so the question was, I mentioned that we're trying to adapt current gene therapy vectors for DMD. How are we trying to do that? And is any of this new work applicable to FSHD? So yes, some of it is applicable to FSHD. We're really doing 2 things to try to improve the DMD vectors. One is to modify the delivery vehicle itself, these AAV delivery vectors. And actually, a lot of that work has already come out of other laboratories across the world. There's been a brand new class or modified version of these AAV vectors that are much more potent at getting into muscle. So the challenge really here is that we're delivering these gene delivery vehicles right into the bloodstream. And the reason for that is that you need to treat muscles all over the body, and every muscle in the body is intimately bathed in blood vessels. So if we can get things into the blood vessels, they'll leak out of those blood vessels and enter the underlying muscle tissues. the efficiency is kind of low, and there are some new classes of these vectors that are up to 40 times more potent in doing that, which is going to allow us to bring the dose down and I think increase the safety. The second thing we're trying to do is we've come up with a new way to co-deliver 2 different AAVs and have them come together within muscle cells and make proteins that are twice as big as ones we were able to deliver previously. And that's important for a variety of muscular dystrophies where you have very, very large genes that are difficult to deliver otherwise. The increased potency of these AAV delivery shuttles is going to be applicable to any muscular dystrophy, particularly FSHD, once we're ready to do a gene therapy for that disease. The larger gene size is something that's actually not a limitation of FSHD, though, only for some other dystrophies. Yeah.
Thanks so much.
I want to introduce our next speaker, Seth Friedman. He's from Seattle Children's Hospital and an expert in MRI. And Friends has funded 2 of his projects, and we're really excited about it.
Yep, thank you. Thanks, Premi. And yeah, if I'm in one of the slides, I will look a lot younger the first time they funded the project. Okay, great. Okay, so thank you all for coming in. For many of you, I've stuck in the scanner over the years, so thank you if I told you in the scanner. So Premi asked me to give sort of a exciting title, and this is the most exciting title I could think about here. I didn't watch a lot of Star Trek growing up, but today I'm going to hopefully get you to individual muscles and beyond in the data lake. Let's see. Okay, so this is really a project that over the last 15 years, we've always collected all this data and we haven't really had a great way to aggregate it. And so over the last 2 years now, I've worked with a company at University of Virginia called Springbok Analytics, and Scott McGarty is here to answer a lot of questions, I'm sure. And we sort of realized that all of a sudden we had this tool that we could start to analyze individual muscles and we could start to put all that stuff together. So I'm going to give you a little teaser on sort of what we're learning. So this is the data that we put into the data lake right now. We have a whole bunch of Wellstone data that we've been collecting over the last 10 years. We also added a bunch of data. There was a study collected at Candy Krieger with Doris Lung, who had data at a very short interval, so baseline and 3 months and 9 months and 15 months, that we thought this is sort of the dream of all these trials, is to understand how quickly are things changing. And then we've been collecting Move Plus data, this natural history study where we're getting whole body MRI that's acquired a number of sites. And we put a bunch of different functional measures into the data lake. In many of the individual studies, we collected a bunch of other measures. So Move Plus, for example, collects reachable workspace. We've not yet integrated that, but we will. So this is where we were about a year ago. I reached out to the folks at Springbok and I said, hey, we have these muscular dystrophy scans. Can we process them? And we started to make these beautiful maps of the legs because that's what we had collected a lot of. And now in current state, there's the ability, let me see how I hide my. Now we're able to make these whole body maps so we can get individual muscle data from the entire body. And you can imagine if you want to model reachable workspace, these sorts of things become really, really helpful. And so where we were, just to review, so most of the time when we talk about muscles, we're talking about a volume. And so we're talking about things like how much lean muscle volume is there, how much fat, what's the fat fraction. And we have a lot of other muscle measures that I'm going to kind of touch on today. And so you can imagine here in the middle is, on one end is the distal part of the muscle, one tip, and on the other side of the plot is the proximal end. And so you can imagine you can start to map where fat is spatially along the muscle. So instead of simply having a value like it's 10%, you can see that most of that is distributed along one side. And then you can also start to make these pixel visualizations. I think this is probably, as patients and as a researcher, this is what I've always wanted, is like, let's actually see where the fat is in your muscle and how that's changing. And so I'm going to primarily focus on how you can take part of our data lake and digest it into some meaningful information. So the first thing we did is sort of something that to probably all of you is kind of boring, but to us it was very important, is to try and understand how much do your muscles really change over a short interval. And because Doris had these whole body scans that were from baseline and 3 months, we were able to basically show that not much changes over 3 months, at least in terms of sort of looking at bulk measures. And so this is good. Like, does anyone in the room feel like they've had changes within a 3-month period? Maybe. So we kind of decided, like, okay, let's make a sort of a confidence interval around this. Let's take any muscle that changes within 5% window and see what actually happens. And this is kind of what it would look like. So if you imagine, here's a baseline scan, and then you have multiple time points into the future. This is a calf. And you can see that at the lower left here, you have more black that's occurring. This is a water image. And so this is a patient that's having more fat being deposited in the medial gastroc over time. You can also imagine you can create a numerical value from this. You could create a volume where you say 30% at baseline and 32 and 35. And then you can create a heat map kind of a view of this where you label any muscle that is 5% or more above their baseline value. So now you can imagine that what I'm going to show you is the lower body. And so this is what a lower body picture looks like at 3 months. So what we've labeled is these are 30 subjects from Doris's data set. This was a full range from ambulatory to non-ambulatory patients. These are all the muscles in the legs and the pelvis and the upper hip. And this is any muscle that changed from baseline to 3 months. And so you can see the values here. Anything that's in sort of a darker blue is a low value of fat fraction, 20% or so. And anything that's a much higher value, like yellow, is quite progressed. And so really what we were able to start to see is that even at this 3-month interval, there are some muscles that are progressing, not very many. And this is the whole cohort, remember. So what happens over a lot more time? So this is the same kind of heat map over the study interval. And so on the top line, you can see these are all muscles that get worse, where fat fraction goes up over time. And you can see that at 3 months, there's relatively few. At 9 months, there's slightly more. At 15 months. So this is essentially the disease progressing across the lower body over time. And then the bottom plot, where you can see is the 5% negative. So this would be if your fat fraction got better. And you can see there's relatively few little ditzels within the bottom row, which suggests that this measure is probably not terrible. But there are some interesting phenomena that sort of illustrates why this is more complicated and why in the data lake we have a lot more pages of data, right? We have fat fraction, we have muscle volume, because there are some things that are happening in the deep hip when you start to have some progressive changes in the hamstring and the quads, which is probably a compensatory response. So you can also plot this in some different ways that I think, we're starting to think about, okay, how do we reduce this down to something that a drug trial might use? And if you look at 3 months, you can also compress these values by subject. And so in the second row here, you can see how many subjects are changing at the different time intervals. And so at 3 months, you can see about 2 of 30 are changing in these different muscles. At 15 months, it's about 4 of 30. And at 21 months, it's really 4 to 6 of 30. So within a sample of 30, you're still only having 15 or 20% of an individual muscle change over time. And when you look in the bottom row, you can start to see that sort of where the magnitudes are, that many of them are less than 50, but there's a lot of muscles that are fairly progressed that are still changing over time. And I want to kind of get into the beyond, because this is sort of on a volume level, this is what we're really interested in. But really what we want to get to is like, well, what's happening at the smaller minute level? And so you can see here, so we can also start to make these pixel maps of that. And we can make these plots where on the left plot, you can see that muscle is in red and fat is in yellow. And at right, this is fat fraction. And so what you can see is here's a soleus muscle that only changes about 3% over an interval. But if you look at the fat fraction curve, you can see there's kind of this shoulder that's progressing. And it's very likely that we can start to look at the area of the muscle that we know should change, and we can really boost our sort of magnitude of effect. The other thing I want to emphasize is that there's a lot of different patterns that we're starting to see. And so one of the common patterns that was talked about is like everything goes from the bottom up, fills up like a cup. We certainly do see that in some muscles. There's also muscles like the sartorius down in the second panel that goes from top to bottom. And then you have muscles that go from like the tibialis, the muscle in the shin that goes from the outside in oftentimes. We've seen some muscles go from the inside out, and some muscles where they're sort of not that fatty, and then the whole muscle tends to progress. These tend to be the long, skinny muscles. And so we do think that sort of we're at this point now where we really want to start to model the whole body together as opposed to these singleton muscles. How am I doing on time? Okay. So the other thing that we want to sort of address is, I think, what all of us in the room that are interested in function try and do, which is how does this stuff relate to the function of the patient? And for years, we've all been looking at plots and making plots that look at fat fraction versus function, whether it's strength or walking distance. And indeed, so if this is the tibialis, the muscle that points your toe, and what we find is that when a little bit of fat starts to increase in this shin muscle, you lose strength very quickly. And it makes sense, right? It's a long, skinny muscle, so it doesn't take much along the muscle. But this fat fraction metric is something that we're all really sort of used to using. The challenge is when you talk to someone that does exercise physiology is they would never use fat as a measure. They would use lean muscle volume because that's actually what's doing the work. And so what's really interesting is when you start to look at corrected lean muscle volume, you start to get a lot more linear plots with some of these functional measures, because muscle is the thing that's doing the work of strength or walking and so I think there's a lot of room to take the data that we already have and to transform it into these functional units of lean muscle volume and start to make much better correlations that we can look at change over time. And then the other thing is this also sort of works for MMT. This is, I think, is an exciting thing for all of us that are collecting MMT. On the bottom left, you can see that is ankle dorsiflexion. So if you take all the ankle dorsiflexors, you put them together, you can see nice relationships there. And then this is maybe where we want to go. Like, you know, we have all this data now, which is great, but it also creates a problem that we have all this data now. And what we're able to start to do is really look at how muscles combine to model function. And you can imagine if all you have is like the calves, well, what muscle in your calves walk? Well, maybe the one that pushes your toe off. If you have the thighs, you can start to add in different musculature. You have the pelvis. So you can start to make these much better models if you can get correlations between individual muscles, but really you want to put them together into an integrated whole. And so where we are now is we have a lot more data to add. So as Stephen and Jeff mentioned, we're going to be doing more scans of patients that we're seeing in the Wellstone study. We are going to be doing a clenbuterol study at Kansas and Rochester and here. So if anyone's interested in that, we'll tell you more. We've been analyzing some of the Move Plus data. There's some data collected in Australia. There's some pediatric data. There's some Massachusetts data. So we have a lot of people that want to put data into the lake, which is, you know, a wonderful opportunity. We also want to put in a lot more functional data that I think will be interesting. Like gait analysis is something that we've always talked about for years. And then we really want to get to the point that we have a way that we can take this data and present it to the FDA so we can make a personalized map of a patient. Because for my money, that's going to be more sort of valuable as a trial endpoint than simply boiling it into the sort of cartoon that we often see with just a couple muscles. And notice I've talked to several of you, like how we really get to the end zone of doing deep analytics and making sure that we can squeeze everything out of this data is our next goals. So there are many people to thank. I feel like as I was pulling little things here, there's a lot of different groups involved. Certainly Springmark Analytics has been an amazing partner in this, and Friends of FSH for supporting this work. And Leo Wong was emailing me as I was sitting there, and he's like, can you please show people this if you're interested in a reachable workspace study at the UW? This is here. This is exactly. So thank you all so much.
We have time for one question for Dr. Friedman.
Yes, George.
In one of your earlier slides, you were showing fat fraction over time, and in some cases, increased fat fraction is over time is likely because of compensation of muscle usage. So that seems to indicate that exercise, more use, exercise might be useful.
Yeah, I think so. The question is, you know, in the plots where I showed things getting better, there is this sort of suggestion that in some muscles, you can see fat fraction go down. And so the question is exercise useful? And I think absolutely. I think that unlike Duchenne's, I think that compensatory exercise and compensation is the thing that as patients you all live with all the time. I do think that the other cautionary tale is that atrophy will also make fat fraction go the other direction. And so, you know, I think this question of, are we seeing hypertrophy? Are we seeing atrophy? What is the other component of the fraction is going to be super important. And we now have all this data in a giant matrix. And so I think we can start to answer some of those questions.
That also implies that imaging will have to be part of any study, because you can't just probably can't just follow like other biomarkers and blood-based biomarkers, because you want to know what's left of the muscle.
Yeah, I mean, I think like, I mean, I'm an MRI, like I put that. So the question was then, shouldn't MRI have to be in every trial? Because then we really need to understand what's happening in the muscles. And thank you, George, for that planted question. I mean, I have a strong bias towards trying to measure what's happening. And so I think I, for years, have sort of felt like we don't know what's happening because we can't make these measurements. And so, I mean, maybe there'll be a point where you could, you know, there's only certain muscles that you really need to measure if you know that those muscles are vulnerable. It seems like as a biomarker, it's expensive, but it's not as expensive as other things, and it's very comprehensive in what you can see. So I'm still betting on MRI. I think we have to, yeah.
Thanks.
Thank you.
Thank you.
Please give us 2 minutes to set up the next speaker.
Please, you know, ask Seth more questions during the silent auction and during the auction. The next speaker is Tessa Arends, and she is a postdoc at Pripatch in Stephen Tapscott's lab. And we have also funded Tessa, and you know, we really want to fund up and coming scientists in the FSHD field and make sure that we have new scientists in the FSHD field. So we are really excited for Tessa to present today.
Hello, everyone. Thank you for having me. So I'm not a clinician, I'm a PhD, and I'm going to take you really down to the molecular aspects of FSHD. I don't know why it went by itself. So I'm really interested in the consequence of DUX4 expression in muscle cells and really trying to understand after DUX4 gets turned on, what does it affect that leads to the toxicity in muscle? And so all of you are aware that DUX4 was found to be the causative gene for FSHD. And this is just showing you muscle in a non-FSHD individual, or those with FSHD1 and FSHD2. So the muscles are colored in green, and DUX4 protein is colored in red. And so you could clear that DUX4 is on in FSHD muscle cells. But what's interesting about DUX4 is that it's rare in its expression, but there's a memory of that DUX4 expression. So here, let me see if I can use my pointer. So here we have a rare nuclei in the myotube that will have a burst of expression of DUX4, and then that will travel down the myotube to affect all of the nuclei, but then DUX4 turns off. However, the nuclei that were affected by DUX4 expression retain a memory of that, and that's really due to what DUX4 turns on. Okay, and so the Tapscott lab and I'm interested in understanding, how does this memory potentiate DUX4 expression, the toxicity that happens later on? And so we found out after that we identified DUX4 as a causative gene for FSHD that it's actually on, as Stephen mentioned, in the early embryo. And here I'm showing you the 2-cell and the 4-cell stage and 8-cell in the human embryo at this cleavage stage. And DUX4 is necessary for driving these events during early embryogenesis. And why is that? As Stephen mentioned, it's a transcription factor, we'll get into that, but for those who are not molecular biologists in the room and need a refresher, so this is the central dogma of biology, and so we have DNA, which has the genes on them. So as DUX4 will turn on a gene in the DNA, and it transcribes those genes. So I always think about DNA as like a cookbook full of those recipes, right? And it transcribes that DNA or those recipes into RNA, which is the recipe, one of the recipes, right, in the genome. And then that RNA is sometimes translated into a protein. So in this case, the recipe was for bread, and so it's made into bread. And so DUX4 is kind of in the stage between DNA and RNA where it transcribes a lot of genes that are really necessary for early embryonic development. But it also turns on, as Stephen mentioned, really repetitive genes, really repetitive sequences in the genome. And I'm actually really interested in one of those repeat elements. That's called HSAT2, human satellite 2. And so normally in the early embryo, DUX4 turns on these embryonic genes and these repetitive elements, but it's fine, right? It's necessary for that expression to happen for the progression of embryogenesis. However, when DUX4 turns on in the muscle, that's not the case. And in fact, DUX4, after it turns on its targets, affects many things in the cell. That includes transcription. Stephen mentioned to you, it turns on its program, but then it shuts off the muscle program. It can affect RNA metabolism, so it can affect how those recipes are read and understood. It can affect translation and proteostasis, so trying to figure out how the bread stays around and doesn't go away. It can also induce cellular stress, so the cells really freak out when DUX4 is on, and it causes a lot of damage in the cell. But that's after DUX4 is gone, right? And so this is where, the lab comes in to really try to understand the molecular mechanisms really governing that toxicity downstream of DUX4 expression. And so there's a lot of us here who study different aspects, whether upstream regulation of DUX4. So a lot of therapeutics are targeting a lot of these things, either DUX4 itself or turning it off. Some of the epigenetic pathways that are regulating DUX4 expression, some of the signaling pathways. So a lot of the therapeutic strategies are really targeting the upstream signaling and trying to turn DUX4 off. We're also really interested in the downstream of DUX4, right? DUX4 is just quickly turned on, but that memory of DUX4 lasts. So maybe if we can also target some of these downstream things that DUX4 turns on, we can also help lessen the toxicity in the cell. And one of these things that turns on again is that HSAT2, that really repetitive element. So what is HSAT2? So HSAT2 is human satellite. It's found, so this is a chromosome right here. And the two chromosomes are linked together, right? At the centromere. And HSAT2 is found in the pericentromere, right below the centromere. And it's silenced in most tissues. HSAT is not turned on. It's found in really, really dense regions of that genome. It's packaged up really tight in what's called heterochromatin, and it's turned off. And when you unwind, when you can see it, it's made up of these repeats. So I just showed you CATTC, and that's repeated forever. CATTC for thousands and thousands of base pairs, 10,000, 30,000 of those base pairs. And here I'm showing you, these are actually human chromosomes. And this is just the pericentromeric region, as you see here in a dark color, really, really dense chromatin. And you have these huge blocks of these HSAT2 repeats. Again, it's normally turned off, but DUX4 likes to turn it on. And this happens in the early embryo. And so here again, I'm showing you DUX4 expression at this 4-cell stage in human early embryogenesis. And we see we have this really nice expression of HSAT2 that correlates with DUX4 expression in the early embryo. We also can show that in an FSHD patient myoblasts or in myotubes. Here I'm showing you HSAT2 in green and DUX4 in red, that we get expression of HSAT2 in FSHD muscle cells. And so what's the consequence of that? Why do we care about HSAT2 expression? It can do a lot downstream of DUX4. HSAT2, when turned on, forms single-stranded as well as double-stranded RNA. And this is not translated. These stay around as RNAs in the cell. So these are toxic repeat RNAs. And they build up. And so here I'm showing you an image of a nucleus. And I'm staining for HSAT2 RNA. And they're huge foci in this nucleus, right? It takes up a lot. And so we have expression of HSAT2, huge accumulations of these HSAT RNA foci. And what that does is a lot. So HSAT2 DNA, because it's highly repetitive and it's huge blocks in your genome, can actually sequester and pull away, kind of mop up a lot of the proteins that are working in your cell and render them non-functional. The RNA can also do that, take away some of these proteins in the cell from doing their work. It can also initiate and kind of keep on DNA damage. And so that's terrible, right, for your cells. You want to be able to repair the damage. But when HSAT is on, it kind of impedes that response. It can also affect RNA processing. So it can impact how those recipes are edited, right, into making new proteins. And it really contributes to the cell death that we see. So when we can deplete, remove that HSAT RNA from those cells, you can rescue, right? You can lose the toxicity even when DUX4 is on, right? If we just remove HSAT RNA, we can rescue that. And so that's what I'm really interested in, is trying to understand maybe how, combinatorial therapies, how we can target upstream to shut off DUX4, but even when DUX4 has already been on, how can we target some of these things downstream, right, in order to help these cells live. And so with that, I'd like to thank my lab, obviously my mentor, Stephen Tapscott, and everyone involved, and obviously with the generous funds from Friends and supporting me in my postdoc career. And I'll take any questions.
You make a distinction between single-strand and double-stranded RNA.
Yeah.
And, is the double-stranded just hanging around longer, or is it more toxic?
Yeah, so they're both toxic. So, oh, sorry. Yeah. Is the single-stranded, how does the single-stranded and double-stranded RNA come about? And is the double-stranded RNA more toxic to the cell? Both are pretty toxic. What's interesting is that the single-stranded RNA and the double-stranded RNA versions of HSAT can sequester unique proteins. And so it might impact different pathways in the cell. And how it forms into a double-stranded RNA is that DUX4 turns on its forward and reverse strands from either side of the DNA. It can turn on the single strand and on a forward way and reverse. They complement each other and they can bind. And then that forms the double-stranded RNA. And so they're always around. They're very stable. These RNAs last for days in the cell. So it's really a long-lasting thing in muscle. Yes.
Is this only in muscle cells, or are all types of cells affected?
So the question was, is this only in muscle cells or are all types of cells affected? And is that in the context of DUX4 expression? Yeah, so we also look at cancer, DUX4-driven cancers as well. And we do see that it turns on HSAT2. HSAT2 also accumulates in those cells and sequesters proteins. So it's really unique to the HSAT, but in all DUX4 expressing cells, we see this happen. Yes.
Do you have a sense of how the HSAT heterochromatin is forming and why you're only getting transcription of the repeat region with DUX4 present?
Yeah, so we know that when you turn DUX4 on, you get complete remodeling of the chromatin. And it's kind of going back to this really early embryonic state where you have a lot of the relaxed chromatin around, opening up a lot of these regions that need to turn on. So that's one of these ways. We're losing a lot of the epigenetic silencers here at HSAT. I think one way is by DNA methylation. You're getting loss of DNA methylation at these sites and activation. And so it's really the remodeling done by DUX4 that's allowing expression. What's interesting is each SAT is on many different chromosomes and we see expression only from a few. So it could be specific to some of these regions of the genome.
The RNA DNA, excuse me, the RNA single cell strands.
It is, absolutely. It's definitely inducing an innate immune response. So when we remove that too, we lose that innate immune signaling.
The online one. Thomas, do you want to ask your question?
I would. Can you hear me?
Yes.
A great talk. Thanks a lot. So I'm puzzled what your ideas might be about delay in this phenomenon. And that is so many people with FSH go for years with no symptoms at all, and they may not develop symptoms until they're teenagers or young adults. What do you think is happening? There's such a major delay in the onset of the disease. Is it simply going on all the time, but very, very tiny levels? Or is there something that happens after decades?
Yeah, I mean, I think that's an ongoing question in the field between us, basic science researchers and the clinicians. And, we have this kind of hypothesis that it's a smoldering effect where you have activity of DUX4, but you really need that ramping up of activity, that memory of DUX4 more and more throughout those years to really have an effect. But that's an ongoing aspect, an ongoing investigation with all of us, is really trying to understand, why DUX4 expression is so rare, why, how many bursts of DUX4 do you need in order to really affect, a muscle cell. So definitely, that's a great question, but still ongoing. Thank you.
And we'll have a 6-minute break.
Yeah.
2:45.
Yeah, please come back at 2:45. Thank you.
Yeah.
I'll unmute it now.
Some like. The nice day out.
Yeah, it was funny.
Looked out with the weather.
Yeah.
Oh. Some of my update. Our next speaker is Jeff Statland, and he's from University of Kansas Medical Center. And he's going to talk about the CTRN, and Friends has funded CTRN for a couple of years now, and we're really excited to see what he has to present this year. Thank you.
Can we get it back to the start?
So I think if we do this and then just work with that. Let me, oops.
Yep.
I see.
Yeah.
Let's do-
It's match for now.
let's see.
Can I control it for me?
Yeah.
Yeah.
So
All
now
right.
you can do that.
All right, great. So what I want to do today is give you guys an update on a clinic-based protocol that we call MOVE that has very open inclusion criteria. And a blueprint of this study was originally funded by Friends, and it's come a long way since that original funding. The power of a study like MOVE, it allows us to answer questions about the design and conduct of clinical trials, about people with FSHD at any stage of the disease, whether they're children or adults, whether they're just slightly affected or unable to walk. It helps us, guides us in making our care guidelines for how we take care of people and understanding how we can improve those. And it's really a platform for thinking about post-marketing surveillance in that time period, hopefully in the not too distant future when we have drugs on the market. When Friends originally funded us, this was back now 3 to 4 years ago. And a lot's happened since then. The pandemic came, you know, the whole nation closed down, but I'm happy to say that we now have 18 centers in more than 3 countries active in this current study. So MOVE stands for Motor Outcomes to Validate Evaluations. It's an observational study. It was a follow-up to a study you may have heard of that we did called Resolve, which was a trial preparedness study, but it allows us to take some of those individuals and follow them for a longer period of time, up to 5 years so far. It includes all people with FSHD, as I mentioned, from pediatric to adult, mentally affected to people who can no longer work, and then genetically walk and genetically type 1 and 2. It's a clinic-based protocol that allows standardized evaluations and care across the centers taking part in the study. It can be performed around those clinic visits, but it has both patient-reported and evaluator-measured functional performance, as well as items that we may pull from the health record. It's a common protocol that allows us to add other things like pilot studies or sub-studies. And indeed, we've had studies of remote assessments MRI and biopsies, wireless motion devices, and we're planning to have a dedicated pediatric study that will be starting in June of 2024. And as I mentioned, it could be a model for post-marketing surveillance.
This is like, volunteers. I think we need to do that. Okay,
Yeah.
Is this the right one?
Yeah. All right, the goals of MOVE, it's to tie your performance now to hard motor health or social outcomes that occurred during the conduct of the study. This would be things like need for braces, mobility aids, breathing aids, or the ability to maintain a job. It helps us understand risk factors for different outcomes, whether it's favorable or unfavorable. This can help us define what a meaningful change would be for our clinical trials and also help us understand our care of patients. It helps us measure the rate of change in different subgroups of people for trial planning and understand the participant-reported measures of change. You can see the different things we collect at each visit on this table. And all I'll say is it really goes from the molecular up to the whole person. Blood for biomarker work, strength and functional measures, and then patient-reported measures of where the disease is at.
I think when there's an admit, yeah.
Okay. All right, the funding for a study like this, it really goes back to this old saying, you know, it takes a village to raise a child. So we have funding from Friends of FSH, but we also have funding from advocacy groups, from the NIH, and from many of our industry partners. So where are we at today? Well, we originally planned to enroll 240 individuals, mainly in the US, but due to interest from foreign sites to join the study, we have close to 300 individuals enrolled. 150 people have completed 1 year of follow-up. 70 individuals have completed their second year of follow-up. You can see the histogram showing the age of the individuals who are enrolled, and you can see it's orange all the way across from 5 years of age all the way up into the 80s. So we have individuals across the full span of the lifespan. We have mostly people with FSHD1, about 5% have FSHD2, which sort of matches what you'd expect to see in a large population. They're mostly Caucasian and non-Hispanic, but we're adding sites now to try to get more diversity into the study. Largely a well-educated group of people, 75% had college or beyond, and about a third of individuals have had their job affected by FSHD, either modified, they've had to do early retirement, or take disability. I want to give an example of the power of a study like this to develop new outcome measures. In this case, one we're sort of excited about. One of the things we've been missing in FSHD is we really don't have a functional rating scale. And so we have a new item, we call it the current ability scale. It's a patient-reported functional motor scale. So this means it's not something a physician has to determine or an evaluator has to measure. It's something that you can report and we can follow over time. If we can validate it, we can use it for following people over time in registries to help guide our inclusion and exclusion criteria for clinical trials, for stratification in those trials, or guidance regarding health outcomes in our clinics. A simpler version of this is in the US registry that's run from the University of Rochester. So if any of you are in that registry, you've probably seen some of the questions before. This was strongly predictive of the need to use a wheelchair over about a decade of follow-up for individuals in the registry, where people who were more severely affected using this outcome were about 8 times more likely to need that assistance. And the categories that were chosen for this match patient-identified prevalent and impactful symptom categories from a large survey that we did. This is an example of the scale. You can see the different areas from the face to the arms. And when you think of function, it's things like combing or shampooing your hair, applying makeup, brushing your teeth. Hand function, for leg function, it's things like walking, running, climbing stairs. And for mobility and transfers, things like getting out of a chair or getting out of bed. And it's the way that you do it that gets the score on the scale. You can see the distribution of scores on the left with the score along the bottom and the number of people who have those scores on the bars. And you can see that the population currently enrolled are mostly mild to moderately affected, with about 10% of individuals falling in that severe range. You can have someone fill this out 2 times in a very short period of time, and you get almost the exact same number, which says the reliability is quite high. You can then take this questionnaire and compare it to other scales that we're already using in clinical trials. Some of you may know about the clinical severity scales if you've tried to screen for any of our current clinical trials. And on the top, you can see the comparison of this patient-reported item to the clinical severity score, and you can see there's a strong linear association. On the bottom, you can see it also has a nice association to the strength that we measure over 26 different muscles in your body. And so what someone's reporting about their functional abilities really correlates well to the strength that we measure in person. We can see changes over time, and we see a similar rate of progression here to what we had seen in the US registry, and it interestingly matches the rates of progression from other types of outcome measures that are measures of strength or function, which is in that range of 2 to 5% per year. By 2 years of follow-up, we're seeing about a 7% change, which on the overall scale wouldn't be more than a point, but still matches, like what I said, what we tend to see when we measure people in person using evaluators or our more sophisticated instruments. The predictive potential of this will be evaluated when we have about 5 years of follow-up, because the other piece of this study that's going to be important are capturing those hard events that happen in someone's life that we can then tie the changes to these other measures to. And you can see a list here of some of them that we're tracking, things like needing a brace, a wheelchair, or non-invasive ventilation. The red numbers that you see in the columns are individuals who are at risk for needing that, who then develop the need for that item over the course of either 1 or 2 years. So far, we've had 27 hard motor events. This basically matches the rate that we had estimated, which was we thought for any one item, we'd see about a 2% rate of events per year, and that's what we're seeing. So by 5 years, there should be enough events to let us to do some analysis to see things like if they're cutoffs on our motor tasks that we measure in clinic that we can use to help you understand what the risk would be that you might need one of these devices in the future. But you can also think about it a very different way. A data set like this can also help us evaluate behaviors that may minimize the risk of these outcomes. For example, exercise or the use of supplements. And that's something we've really never had a data set to do before. So as we think about our future directions, we have 18 trained sitters with several who are set to join this year. We have pilot studies that are running now, including remote motion analysis using the SysNav system that some of you may have heard of, and a pilot of whole body MRI for the children. We have sub-studies that use the MOVE protocol to build on, including one where we do whole body MRI in reachable workspace, and a dedicated pediatric study, like I said, that should start later this year. And what our real goal is to expand and maintain this cohort. I think the real power here is the longer we can continue follow people, the more powerful this kind of data set becomes. And then it becomes a model for once we do have treatments available to try to understand how it's affecting people in their real lives as we move forward in time. All right, thank you very much. I'm happy to take any questions if there's time.
I have a question. Can you tell us a little bit about what the patient experience is like participating in MOVE? How often do they come into the clinic? Are people traveling from where they live to come to one of the centers, in case we have folks who might be interested in participating?
Yeah, so for the average MOVE visit, it's designed to match the time you're going to be seen in clinic. We have a lot of centers that are participating. And so it would be on the time when you normally went into clinic, they would collect a lot of the pieces. Now, some clinics will have the resources to do all of the assessments in that clinic space. Other institutions may have to take you over to their research space to do some of the assessments that we're collecting. It probably takes about a couple hours to do your actual clinic visit, plus then everything else that we do. Now, some of the sub-studies have a little bit more involved, like the whole body MRI or the reachable workspace. For those studies, there is some funding there to bring people into centers if they're interested in participating in them. And then when we start our pediatric study, because some of the children, it's hard to catch them when they're young, we're going to have money to fly those people into the centers. Yeah.
Excellent. And patients are usually doing it once a year then?
Yeah, so the MOVE is once a year. Some of the sub-studies may have slightly more frequent visits, depending on what the aims of those sub-studies are.
Thank you.
Yeah.
Other questions? Stuart?
So anecdotally, you hear stories of periods of stability, periods of not decline. Does that show up in your data at all?
Yeah, I mean, that's a very good question. Yeah, all right. So he's asking, we anecdotally hear people, and I hear this in clinic all the time. People will talk about stability and then these periods of very rapid change in their bodies. You know, when we look at these large data sets and you look at something like strength, really what you're struck by is you see a sort of slow incremental change over time that's somewhat linear when you look at the whole population. It's very difficult to catch someone exactly at that time when they're having that rapid change. Now, some of these larger data sets, we may be able to do that analysis. But, you know, up to now, like I said, it's very difficult to do that because you have the normal variation that you have in some of the ways you measure things to account for as well on top of that.
Do you have, I think that you cannot be part of a drug trial to be part of MOVE now, is that correct?
So the question about drug trials and MOVE. So MOVE, we're completely happy if you're in a drug trial, if you want to be in MOVE, but the inverse may not be true. And so what you'll need to check is see if the drug company would be okay if you were in the natural history study.
And I had a follow-on question. It was regarding exercise and nutrition supplementation. Is there anybody going or doing anything in terms of prescription as to what's recommended and how much of the different aspects there?
Yeah, so the question was whether anyone's doing anything to give a better guidance about the amount of exercise and the kinds of exercise or supplements for people to use. There's a lot of interest in this. FSHD, probably more than any other dystrophy, has actually had a lot of studies of, you know, moderate to intense aerobic exercise. So I think one thing we can say is it certainly isn't going to hurt people to be doing moderate aerobic exercise, and it's helpful for your cardiovascular health. As far as resistance exercise, there's a little less data to inform it, but it looks like it probably is not making people worse. And so you can build it into your routine. Right now, you're right, many clinics do give different recommendations based on the providers. I think we're currently making some guidelines to try to have more of a centralized approach to this. And those guidelines will hopefully be out in the next year.
You can take one more, go ahead.
So are those, would those guidelines be applicable or available to primary care doctors who are taking care of these patients, because a lot of people don't have necessarily easy access to a center? And so they're going to someone that may not have much knowledge and where do they get their guidelines?
Yeah, so the question was, if there are guidelines, are they going to be available to primary care providers? That's a great question. I mean, the answer is yes. Like, once the guidelines are there, they'll be posted and they won't cost money for people to get them. The part that's a little trickier is the education, so these providers understand that those guidelines are there for them to download. And we probably need to be doing a better job of getting the word out that way. And yeah, or the patients, you guys can certainly help us get the word out. Yeah.
Okay, let's thank her for Seth.
Yeah, thank you.
So, Robbie Toil is here and Jeff and his team are here. And so please make sure to talk to them face to face during the silent auction and so on, because they're, I think might be the first time that they're here. So, yeah. And you have, so I'm going to bring this up. Miracle. Our next speaker is Anthony, and he's the CEO of Miracle. We're gonna hear the research progress from him. Let me share my screen. All right. Yeah, and then you can control from here.
Yeah, thank you, Premi, for inviting me. I'm Anthony. I'm CEO and co-founder of Miracle, and we're lucky enough to have received a grant around the time we started our FSHD program for Friends of FSH, and I think research, and really happy to have the support and work with everyone here on our program. We're also lucky enough to be partnered now with Sanofi on that program, one of the biggest pharmaceutical companies in the world, really well known for rare disease and developing disease, especially for developing therapies for neuromuscular disorders like Pompe disease and other rare diseases. Hopefully they'll be able to help us bring this drug to as many patients as possible when we bring it forward. Like I mentioned before, we're focused on RNA therapeutics, and I thought it might be good to take a step back and talk a little bit about the basic biology, how RNA therapeutics work, and how we hope our treatment will work for FSHD. So, and I think a lot of the speakers earlier before to me today have already talked about this, but I'll go over it again. As many of you may know, our genetic information is all stored in 23 sets of chromosomes on our DNA. Every cell in our body has a copy of those. And each chromosome is two very, very long strands of DNA. And regions of that chromosome are organized into what we call genes. And that contains an area that controls that gene to come on and off. Your chromatin opens up, and then you expose this double-stranded DNA. Each of those two strands is a complement to each other. So one strand has all the information to replicate that other strand, and that's how our DNA replicates. That's also how we express information from every gene in our body. And I'll tell you a little bit about that. Oh, I didn't have my animations going. So we have our DNA, right? And each of the region of that DNA encodes a gene, right? What happens when information from a gene is used by the cell to perform a function or to replicate is that opens up and you transcribe a messenger RNA. So that RNA is complementary to one of those strands. And the way to think about RNA is it's that temporary message. The DNA is like your hard drive in a computer, or etched on stone, the permanent information. The RNA is the intermediate information that's going to RAM, and that gives you that message that uses that temporary information to construct what are proteins in a process called translation. And those proteins are like little machines. They're made of a different type of material, things we call amino acids, and they can perform all the functions in the cell. And that's how all the functions in our cell and in our body happen, is mostly through proteins that are encoded from our DNA. When we inherit a disease often that causes an underlying mutation in the DNA, and then that changes that sequence. And when that sequence changes, you get the wrong information in that mRNA, in that intermediary, and then you get the wrong information in that protein. You have a dysfunctional protein. Most of the diseases we have, they're either inherited from our parents or it happens spontaneously. That's what happens in FSHD. That's what happens in cancer. You get these spontaneous mutations, and that messes up the normal function of our cells. And the way RNA therapeutics work, well, maybe I'll take a step back and talk about first for FSHD. On one of our chromosomes in our body, chromosome 4, we have a repeated region where the DUX4 gene is. A lot of people have talked about this today. And the mutations that we get, we lose some of the DNA that controls expression of the DUX4. You still actually have a normal copy of the DUX4. As Stephen's group said earlier, that's normally only supposed to be expressed very early in embryogenesis. So for 95% of us, we get that mutation in chromosome 4, we lose the regulation, and then DUX4 comes on in our muscle tissue. That becomes cytotoxic to that muscle, and slowly over time, you get atrophy of that muscle. And so what our company is doing, and other companies in the space, is we're trying to develop an RNA therapy that will specifically target that intermediary. So as your DUX4 gene comes on in your muscle cell, you're making this mRNA, and we'll make a small RNA that binds to and destroys that DUX4 mRNA, and then that will eliminate expression of the DUX4 protein. If we can do that successfully, hopefully we shut down specifically that expression in our muscle cells, and those muscle cells go on to normal function, and then hopefully our muscles don't atrophy, they stay in normal shape as we age and go forward. Now, the issue with RNA therapeutics, the big challenge is that you have to get them inside cells. Our cells are perfectly designed to keep foreign nucleic acids out, because otherwise that's how viruses come in and they infect those cells. They have to figure out how to get across that cell membrane. And so if we just inject RNA into the body, it won't go into the cells, won't be able to go in and target that underlying genetic program. And so what we've done is we've developed a technology, and this is where our partnership with Sanofi comes in, to use an antibody. So these are natural proteins that we all express. And so we make an artificial antibody that binds to a receptor on the outside of the cell. And then that allows that RNA to be bound to the outside of the cell, and that asks the cell to bring that RNA in. After it comes in, it gets separated from the antibody and specifically target and destroy that DUX4 mRNA and eliminate it. Now, there's some special things about the antibody technology we're using, and that's where the partnership with Sanofi comes in. So Sanofi owns a company that takes antibodies not from humans, but from camels. And the reason why they focus on that is those antibodies are very small, less than 1/10 the size of an antibody that we express. And the reason why we want to focus on that, those small antibodies can be administered better, but they can also penetrate tissues better. So as some of the other speakers talked to you about today, DUX4 is expressed in 1% of the cells in our body, but you have to, so that means if we want to shut off DUX4, we have to get almost every cell in the muscle in order to shut off that 1% that are expressing DUX4. And by using these smaller antibodies that can distribute through the tissues better, hopefully we'll be able to get all of those cells and then have that antisense RNA, that anti-DUX4 RNA there, knocking down the DUX4 protein. And so the first question you want to ask is, can we actually do that if we build one of these, conjugates that has the antisense RNA and the antibody, does it get taken up by cells? And so what we do is we take cells from patients, their myoblasts, and we differentiate them into something that's like a muscle fiber. It's not exactly a muscle fiber. We do that in a dish. If we label our therapeutic with an infrared dye, we can see if it's going into the cells. And so we can see here, we're hitting all of the cancer cells in a dish. So that won't tell us what's going to happen in the body, but at least starts to show us the therapeutic can work. And then the second thing is, if we do that again, now, and we collect those cells and we look, is the DUX4 RNA being eliminated from those patient cells? So there seems to be a little bit of a problem with the, so I have a figure in here that you can't see on the slide where we're actually collecting RNA from the cells and we're looking at the DUX4 RNAs getting knocked down by those conjugates. The next thing I want to talk to you about is how we're going to administer this drug. And so, as many of the other speakers have talked about, FSHD doesn't happen across every muscle in the body. It seems to come up in certain muscle groups at certain times. And one way to administer drugs is subcutaneously. So you put injection on the skin and that distributes around the body, but you always get the highest concentration of the drug near where you inject it. So if you know anyone who has type 1 diabetes, they can administer that insulin to that patient subcutaneously. And we want to try to do the same thing for FSHD. And so what we're looking to do is we have these auto injectors that you can put on the skin at different locations of the body, and then they'll infuse that drug into the patient, injecting it under the skin automatically. And if you do that, you can place that auto injector near in certain locations on the body, near where maybe the most affected muscle groups are. So if you have, shoulder involvement or lower leg involvement, we can try to place the auto injector there so you have the highest concentration of the drug near where those muscles that are most affected. We don't know that we would always want to administer the drug that way, but it gives us the opportunity to try to target the treatment a little better to where the patient needs it most. And then lastly, just showing you a little data, in an animal model, can we get these therapies to work? And so what we've done here is we've taken one of our conjugates and we inject it sub-Q like we would on a patient into a mouse. And then we place the label again on it so we can see where it goes in the body. So within a day, it's distributed all over the body of the animal. But if we pull out one of those muscles in the leg, we can bring it up. You can see on the top, that's looking at the overall infrared signal from the muscle. And below it, we're seeing, just the infrared signal for the antibody. We're seeing broad distribution through the leg muscles and uptake. We've also done studies where we've looked again at DUX4 expression in those tissues and can see that we're knocking down DUX4 in those muscles and bringing them down. So hopefully in the coming years, we'll be able to bring that drug forward, and we'll be able to start testing it in patients and seeing if they can, if this therapy we're developing can actually correct that underlying DUX4 expression in muscles directly. So I want to thank our collaborators at Sanofi that have been, we research closely with, that are developing the antibody portion of our drug, and our team at Miracule developing the RNA therapeutic, and our supporters like Solve FSHD, which invested in Miracule, Friends of FSH Research, and the FSHD Society.
How long does it hang out in the blood, the antisense?
So the antisense drug itself is very, very stable. It doesn't get broken down very quickly. So once it gets taken up by cells, it's maybe weeks. But when you administer it sub-Q, it gets in the blood, and within a couple of days, hopefully it's gone from the blood to the muscles. And so you have this rapid peak like in the first day or two, and then it comes back down. But it hasn't been eliminated from the body. It's gone from the blood to the tissues where it's being uptaken and then retained for a much longer period of time.
It's a shot, periodically, not a infusion.
Yes, that's the whole point, is you would subcutaneously administer it, and it would diffuse from that site around the body of the patient. And so that would happen maybe once a week, once every few weeks, that administration would happen.
Anthony, wonderful talk. I'm wondering what we know about long-term tolerance and drug antibody defense.
So that's actually a really good question. And oh, yeah, sorry. So the question is about if we generate immune reactions against a foreign antibody, specifically those antibodies from camels. And part of Sanofi's technology is what we call humanizing those sequences. So we take the sequences that are foreign to humans for the structure of their protein, and we put human-like sequences in that the human body sees as natural. And that took years and years of research for them to identify a structure where they could modify out the camel sequence, modify in human sequence that allowed that antibody still to be functional.
Thank you. With this system that you're with subcutaneous, where are you in the stage? Are you like experimental, prototype, like?
Yeah, I mean, so we kind of classify drugs. So we definitely have proof of concept, and we're still optimizing some of the features of the therapeutic in the preclinical stage. So modifying the structure of that antibody. The RNA structure is pretty well developed, and we think we have the lead compound that will go forward to humans there. We're still optimizing the antibody some to get the right stability, safety profile, solubility of that drug. And so then hopefully then we'll be able to bring it from preclinical through regulatory studies in the next year or two.
PDF. Might have to scroll, I think. Okay. Well, our next speaker is Wes Miller, and he is the Chief Medical Officer for Epic Bio. And we're excited to see what he's going to present here. Yeah.
Thank you, Premi. And again, my name is Wes Miller. I'm absolutely delighted to be here on behalf of the team at Epic Bio to talk to you today about EPI-321, which is a candidate investigational product for the potential treatment of patients with FSHD. So thanks to the friends for inviting us to be here. Before I talk about our platform and as our tech platform and also EPI-321, I just wanted to talk a little bit about our company. We're based in South San Francisco in California. We're a team of 49 people, so relatively small, but incredibly hardworking and dedicated people who absolutely have people living with FSHD on their radar. As a disclaimer, I'm a pediatrician and I have never knowingly clinically cared for a person living with FSHD. But again, I am humbled and really privileged to be able to work with the team at Epic Bio on this program. So before I talk about EPI-321, I think it might be helpful, sorry, I have to scroll very slowly here. I think it would be helpful to talk about our core technology platform at Epic Bio, which we call the Gene Expression Modulation System, or GEMS, GEMS. So GEMS is a CRISPR-Cas-based system, and that allows for us to specifically and precisely target areas within the genome and to selectively and precisely activate the expression of genes. So either activate the expression of a gene, or in some cases, as in the case for FSH, to suppress the expression of a gene. So similar to other CRISPR-Cas systems, we use something called a guide RNA, which is shown in this cartoon as the purple strand. So guide RNA is a molecule that is comprised of RNA, and that is designed or engineered in a way to seek out within the nucleus and the DNA a specific and precise region or target of the genome. And that allows then the yellow blob that you see there, which is called a CRISPR-associated protein, to find the guide RNA and to affect some change within the genome. So together, that purple guide RNA and the yellow Cas protein or CRISPR-associated protein create some effects within the genome. So this is now where our system starts to diverge from the more classic CRISPR-Cas systems that you might be aware of, including those that use Cas9, such as the one that Jeff was talking about earlier that was recently approved in a product called Exa-cel for patients with sickle cell disease. So more classic Cas9 proteins are nuclease active, which means once they find the guide RNA and bind to an area in the DNA, they create a break within the DNA. This damages the DNA. our protein is engineered to be nuclease dead or deactivated. So therefore we call it DCAS. And so while it finds the guide RNA and binds specifically to the targeted intended region, it does not create a double-stranded break or even a single-stranded nick within the DNA. Instead, that Cas protein is engineered to be bound or fused to what's shown as a blue circle, which is an effector protein, which then has some enzymatic effect on the epigenome in order to either activate or repress a gene. Oops. So how does this apply to EPI-321? Well, you might be able to guess. We are intending to use our GEMS platform for its suppression activities of genes or a target gene by way of modifying the epigenome. So in this cartoon, I am showing you exactly what others before have talked about today. On the top level is a hypothetical normal DUX4 region on the 4th chromosome. And so this is stylized in this cartoon, and it's somewhat oversimplified, but it's showing an expanded D4Z4 repeat unit length next to the DUX4 gene. And that region is well methylated. So the methylated groups are shown as those green circles. And because of this extended normal repeat unit length of that region, and because it is very well methylated, this shields that region of the DNA from transcriptional activity. And so DUX4 gene remains pretty much silent. So we don't get expression of that particular protein. And therefore, in healthy adults and healthy people, they don't get the downstream effects of DUX4 activation or FSHD. The second line is showing a hypothetical person living with FSHD. So this would be type 1 disease in which that region has been shortened because of loss of repeat units within the DNA. That leads to then a change in the epigenetic environment or the chromatin structure, which we know now is also characterized by loss of methylation within that region. And so that's symbolized by the open green circles that you can see there. That, of course, then leads to DUX4 expression. And for the reasons that we've heard about today, that leads to toxicity to muscle cells, muscle cell death, and then the signs and symptoms of FSHD. And so at the very bottom, you can see how we are intending to use our gene editing, GEMS program to be able to shut down DUX expression. So our guide RNA is specifically engineered in this instance to find and locate to the area of DNA on the 4th chromosome next to the DUX4 gene. Our GEMS protein is designed in a way, or our Cas protein is designed in a way to be fused to a modulator that then has a methylating activity, which remethylates the region and therefore shuts down DUX4 expression. So all of that activity of our CRISPR-Cas system is happening within the nuclei of cells. And of course, we want this activity to be happening within the nuclei of muscle tissue. So an important and salient question is, how are we delivering the GEMS construct within our EPI-321 product to muscle? And the answer comes from what Jeff was telling us about earlier, which is that we are using a well-known and well-characterized now delivery or vector system called adeno-associated viral vector. In particular, we're using a serotype of AAV or adeno-associated viral vector called RH74. This is the same serotype of AAV that is used in Elevidys, which is again the recently approved gene transfer product that confers microdystrophin to muscle cells for boys who are living with Duchenne muscular dystrophy. And so this is intended to be a one-time dose of this investigational product. In other words, the intention and expectation is that patients or people who are living with FSHD would receive this product one time. We have good reason to believe that we would be able to get durable effect, but it's also important to know that after a person is exposed to an AAV vector, that it is expected they will develop significant immune memory to that vector, which then likely will preclude them from being able to get this specific vector type in the future. And it also might preclude them from getting other AAV serotype vectors in the future as well. So what does this candidate investigational product EPI-321 look like? Well, again, it looks like overall a protein capsid of an engineered adeno-associated viral vector. Importantly, that AAV is engineered in a way so that its own internal DNA is gutted. So that means that it is not a replication competent virus. It can't create an infection in you after we deliver it to you. And instead, in place of what would be the normally occurring viral DNA, we put in this sequence of information which encodes the GEMS construct. So this part is basically encoding for the guide RNA, which again is the GPS to find that region right next to the DUX4 gene. We then have our dCas onyx, that's the special name of our Cas protein, fused to the modulator, which then achieves methylation once the dCas onyx attaches to the guide RNA next to DUX4. Importantly, our Cas onyx, dCas onyx, and modulator are under the control in this DNA sequence of what's called a muscle-specific promoter. And so this means that while AAV we know can transduce or penetrate into lots of different tissue types, not just muscle, but others, we expect that only within muscle, where it will sense the right information and transcriptional activity, will our GEMS construct be expressed. So we have not begun clinical studies of EPI-321, but we are preparing to submit what's called our IND or CTA to regulatory authorities in order to be able to begin our first in human and first ever clinical study of EPI-321 this year. And so as part of that preparation, of course, we have conducted extensive testing of EPI-321 within so-called preclinical models. And the first that I'm showing you is an in vitro. So this is in culture and lab or Petri dishes model that's studied two patient-derived FSHD type 1 patient-derived myoblasts or primary cell lines. And these were myoblasts that were put into culture. And then when they were in culture, they were exposed to EPI-321. So this is the same EPI-321 that we are planning to use in our clinical studies. So down here, we're first of all looking at how the exposure to EPI-321 changes the transcriptional activity or transcript levels of DUX4. On the left, you can see the control condition. So these are the two FSHD type 1 patient cell lines that have gone into the dish and not been treated with EPI-321. And we've normalized the DUX4 transcriptional activity at 100% or 1. When we expose to a relatively low dose of EPI-321, again, within this culture system, we see that we can measure our dCas onyx. So we know that the product has transduced the muscle cells and that our GEMS construct is being expressed. And we also see that we're able to lower the DUX4 transcriptional activity. At higher doses, we can see a dose-dependent response, meaning we get even further suppression of DUX4 transcription. And of course, we can measure higher amounts of the GEMS product transcription itself. On the right side, we're looking at perhaps what's a more important downstream consequence of suppression of DUX4, which is cell survival. So this is measured with a so-called apoptosis assay, looking at a caspase 3-7 assay. And so on the x-axis, or sorry, the y-axis, you can see the cell survival that's normalized to the negative control. So the negative control, again, is shown in orange. These are the cells when they are not treated with EPI-321. And you can see, again, at low dose or low exposures versus higher exposures, that we get significantly higher cell survival over time. And on the x-axis, that's shown in hours. So in addition to an in vitro model, we've also looked at what changes EPI-321 might be able to make in an animal model and an in vivo model. We have used the humanized FSHD mouse model. So many of you are very familiar with this model likely, and this is where we take, again, patient-derived FSHD type 1, in this case, muscle tissue, and it is xenografted onto immune-incompetent mice. And so we put these into the tibialis anterior region of the mice. This allows us then to give intravenous doses of EPI-321, much in the way that we would plan, or we do plan to give EPI-321 in the clinic. And we can administer them at clinically relevant normal doses, normalized to the mouse body weight. And so again, you can see on the left here, this is a graph that's looking at the change in the human muscle tissue DUX4 transcriptional activity. This is vehicle, which means when we give just placebo and we normalize that to 100%. And then at low and high doses, we see significant reductions in the DUX4 transcriptional activity. And similarly, we can look at apoptosis again on the right side. So that's a measure of cell death. And we can see in the untreated state, this is normalized again to 100%. And then with low and high dose, we can see reductions in the amount of cell death that is resulting from DUX4 transcription. So we are hoping and expecting to initiate our first in human clinical study this calendar year. Because this is an AAV therapy that essentially is using an epigenetic editing approach, we will be conducting the first in human study in actual patients. We will not be studying this first in healthy volunteers. We are planning up to 9 participants in this phase 1, phase 2 study. The participants are going to be people living with type 1 FSHD. They will be adults. They have to have and demonstrate adequate vital organ function in order to be able to undergo AAV therapy. We are planning a multi-center study with likely up to 5 clinical trial sites. This is going to be an open label study, which means that we are not going to be using in this very first in human study a placebo group. All patients who are enrolled and who get dosed will get the active drug product or EPI-321. Like other AAV therapies that are administered systemically, it's given as a slow IV infusion. Also similar to other AAV therapies, patients will be required to take a type of immune suppression, which is a fairly common medication called prednisone or an equivalent to prednisone. And we expect that that prednisone will begin a couple of days prior to dosing and will need to continue for many weeks following, then with a slow taper to off. The key endpoint and objectives of this first in human study is going to be to understand the safety and tolerability of EPI-321 in people who are living with FSHD. But of course, we have many important secondary objectives and endpoints that are looking at the pharmacodynamic activity of EPI-321. That is, does it get into the muscle tissue? Does the GEMS product get or construct get transcribed and translated? And then do we see evidence of DUX4 transcriptional reduction as well as evidence of methylation near the DUX4 region? And so, of course, we're going to need to accomplish that by looking at muscle biopsies at baseline and then at various time points after dosing. We're also planning to use MR imaging, again, in order to understand by that modality evidence of extent of disease and disease activity and how that might change over time. And then we are planning to use various functional outcome assessments as well as patient-reported outcomes that are currently being used in other interventional studies for people living with FSHD, and that also have been used in recent and ongoing natural history observational studies. So hopefully this gives you an overview of our EPI-321 program. I wanted to thank Friends of FSH again for inviting us to be here today, and also just thank everyone in this room who's obviously contributed to our understanding of this disease, which really enables us to attempt to apply in a rational way our platform to try to help patients who are living with this disease. So thank you very much, and I'm happy to take any questions. Sorry.
You don't get a dose-dependent reduction of DUX4, but you get a xenograft model.
So the question, yeah, so the question is, why do we think we don't get a dose dependent in the xenograft model? So very good question. And I can tell you that we have looked at not just, so sorry, did I say the full question? Okay. We have looked at not just DUX4, but some of the downstream genes that of course are turned on by DUX4. When we look at various composite scores that others have looked at, we see a much stronger response. I can't tell you, and we're pondering exactly why we're seeing a dose response in the composite, but not actually in DUX4 itself. It's also possible that we are also hitting a bit of a ceiling effect. So in some weird ways, our AAV administered therapy is fundamentally different from those which are just trying to get a transgene that's episomal, right? And that's just constantly expressing and translating some protein, such as a microdystrophin. So stoichiometrically, we're bound by the targets that we have, right? And so in theory, because we do believe in the precision of our product, those targets are numbered, they're small. And so it may just be that we're getting maximum in this model shut down because we have fully methylated as best as this product can do.
I have a question. Is there a difference in the way a Cas protein can bind to, you know, heterochromatinized region, like with multiple alleles in FSHD?
So the question is there a difference in way that the Cas protein can bind to various different alleles? We don't think so. We have looked, I showed 2 different cell lines for the in vitro data, but we've actually looked at a total of 10. And in all 10, we are able to see in our in vitro programs reduction of DUX4. We also are able to measure change in methylation as well. And so, you know, I guess arguably, if we were able to look at hundreds of cell lines, we might be able to discern if something like repeat unit length might have a difference in terms of those outcomes, but we haven't seen a difference in the 10 lines we've looked at. Yeah. Yes.
Since it's a one-time dose, if a patient got it and then say they exercised and they increased their muscle bulk, does the new muscle tissue could it potentially also have the therapy, or would it?
Yeah, so I think, maybe the question, and tell me if I'm correct, maybe the question is, if a cell is not post-mitotic, if a cell still can undergo mitosis and division, what would be our expectation for a cell that previously has been successfully delivered this GEMS, right? Is that what you're saying in terms of the daughter cells? Yeah, so what's really interesting is that once the methylation is changed, it's imprinted. And so this has been well accepted in the field of epigenetics that if you can get a methylation change in a parental cell that then undergoes, of course, replication of its DNA, that methylation status is imprinted onto the daughter cells that are produced. So, you know, I think we now know from other kind of neuromuscular experience with AAV that AAV persists for a long time. So another thing is that AAV could persist, you know, episomally for quite a while and be delivered to a daughter cell if there is enough around. But even without that, we have some degree of confidence that that methyl change would be, or methylation change is imprinted to daughter cells.
I've got one question for you all right here. Will this treatment work for FSHD?
So we think that the treatment, sorry, the question from online was whether we would expect that this treatment would work for FSHD type 2. And we think that based upon the mechanism of action that it would. We are in the first in human study only planning to enroll patients with type 1, and that's because all of our work has really depended upon patient source cell lines, and thus far that's only been conducted in patients who have type 1 FSHD. Yeah.
Thank you.
Sure. Sorry.
While we're switching presenters, I'm going to pass around the sign-in sheets if you want access to any of the presentations today or recording.
Hi.
Just have you signed up here.
Yeah, I was looking for you. So you have, you're on Zoom. Okay, great. Let me just, and you have, yeah. So we can do this. And yeah, go ahead and put this. It will take a second. Okay.
Oh no, I'm in presenter mode. We get out.
Swap. Is that right?
It's like the bane of my existence. Behind presenter. There we go. Okay, thank you.
Yeah. Yeah, and we can. Exit. Yeah. I can just introduce you. Sure. So our next speaker is Christina Tysoe, and she's a clinical scientist at Avidity Biosciences, and she's going to give us an update on FORTITUDE study.
Hi. First, I want to thank the Friends of FSH Research for inviting our team to give an update on the FORTITUDE study and to talk about avidity as well. So like your kind introduction said, I'm Christina. I'm part of the FSHD clinical development team at Avidity. And before, oh, sure, sorry. So before I get started, this is just the general disclaimer slide that I'm going to be making some forward-looking statements in my presentation today. So first, I just wanted to give an introduction to Avidity. So at Avidity, patients are really at the heart of everything that we do. And our mission is really to improve the lives of people impacted by diseases that currently have limited therapeutic options, including FSHD. And we hope to do so by advancing a new class of drugs. And so this new class of drugs really relies on this innovative technology that we've developed at Avidity. And this innovative technology, we hope will allow us to directly target affected cells while also addressing the root cause of the disease. So it's really important for us to partner with the FSHD community in everything that we do at Avidity, including how we design our clinical trials. And the clinical trial I'm going to be talking about today is the FORTITUDE study for AOC 1020. And this study is currently ongoing. And we're really excited about the fact that we're going to be able to share some preliminary data from FORTITUDE later in the first half of this year. So all of our compounds at Avidity use this AOC technology. And AOC stands for antibody oligonucleotide conjugate. And we've designed a number of AOC compounds that allow us to specifically target muscle cells. So thinking about the building blocks of the AOC platform, the first key component is the antibody portion of the drug. And so antibodies are naturally producing proteins in our body that we use to fight infection. They're really great tools in drug development since you can design antibodies to specifically target specific proteins, including those that are different between different types of cells. And antibodies have been commonly studied in drug development and have a vast safety profile, and there are over 100 different approved drugs using antibodies, including many for chronic conditions. The second key component of our drug is the oligo. And so an oligo is a short strand of RNA or DNA. And oligos are powerful therapeutic agents themselves because they can really allow us to target gene expression. And this is what allows us to target the root cause of the disease. So thinking about AOC 1020 specifically for the potential treatment of FSHD, the antibody portion of AOC 1020 is an antibody that specifically targets the transferrin 1 receptor. And so transferrin 1 is ubiquitously expressed on muscle cells, and binding of AOC 1020 to the transferrin 1 receptor allows for uptake of the drug conjugate into the muscle cells. Once inside the muscle cells, the oligo portion of the drug disassociates from the antibody and is taken up into the it's not RISC complex. I always say RISC complex, but the C is the complex part. So it's taken up into an RNA-inducing silencing complex, and that's what then binds to mRNA of DUX4 and prevents those toxic downstream effects that Tessa was talking about earlier. And this is what we mean when we say that this allows us to target the root cause of the disease. So we've done a number of preclinical studies on AOC 1020. This first study is using the AOC 1020 siRNA in patient-derived myocytes. So when you use the siRNA component of the drug, we see a really nice dose-dependent decrease in that DUX4 gene signature. And we can see an up to over 80% reduction in the DUX4 gene expression. The second study is using that AOC 1020 siRNA component, this time in a mouse model. And again, we see that dose-dependent decrease in the DUX4 gene signature in the mouse muscle cells. The key thing I want to highlight here, though, is that after a single dose of the AOC 1020 siRNA, we see a maintained decrease in that DUX4 gene signature up to 56 days. And this is really promising, and we hope that it means that we'll only need to use infrequent dosing for AOC 1020. So this final preclinical study that I'm going to talk about is in a transgenic mouse model. And so this transgenic mouse model has been designed so that when we give the mouse tamoxifen, it will express DUX4. And that has nothing to do with tamoxifen or DUX4, it's just the way that the mouse model is designed. And you can see that in the mouse model that has tamoxifen-induced DUX4 expression, the mouse really has a decreased ability to run on the treadmill. And also these mice have a decreased ability to produce muscle force. However, if we give those mice AOC or mouse version of AOC 1020 before the tamoxifen, in order to allow for loading of that risk complex, we can show recovery of the ability to run on the treadmill and a recovery of that muscle strength. So next I'm going to jump into talking about the FORTITUDE study specifically. And so FORTITUDE is a first-in-human phase 1/2 study for AOC 1020 in adults with FSHD. So there's a lot of components when we design the FORTITUDE study. The key thing here is that FORTITUDE is a randomized, placebo-controlled, double-blind study. That means that participants will be assigned to receive either a placebo saline injection or an injection or injections of AOC 1020. And both the participant, the investigators, and us at the sponsor will be blind to who is receiving what compound. So given that this is a first-in-human study, the key goal is to test the safety and tolerability of this compound. But there's also a lot of other interesting questions that we want to address, including how well our drug is taken up into the muscle cells and how well it can impact efficacy measurements. So we're going to be doing muscle biopsies, we have MRI imaging, and we have a number of muscle strength, function, and patient-reported outcome measures. So this one kind of repeats what I said on the last slide. So given that this is a first-in-human study, we want to start dosing at a very low dose of AOC 1020. So we have cohort A, which will receive a one dose of 1 mg per kg. And then after some safety review, we will increase the dose level of cohort A before expanding into a higher dose cohort. And so dosing in the FORTITUDE trial is quarterly. We do have one booster dose at 6 weeks, just to really promote that loading of the risk complex, and before we have the muscle biopsy. The other thing to highlight here is that while this is a placebo-controlled study, everyone in FORTITUDE will be rolling over into an open label extension study where everyone will be receiving AOC 1020. So the study is ongoing, and we've had like a lot of really great enthusiasm from the patient community, and we're going to be presenting some preliminary data on the study in the first half of this year. So this slide just summarizes those key things. So we're studying AOC 1020 to potentially treat the underlying cause of FSHD. This drug is administered by an infrequent IV infusion and is currently being studied in the phase 1/2 FORTITUDE study. And we're going to be sharing some preliminary data on this later this year. And again, I just really wanted to thank everyone in the community, the clinicians, the patients, Friends of FSHD for partnering with us to really advance this shared goal of developing an FSHD therapeutic. And so this is just a picture of everyone at Avidity. If you have any questions beyond, if you have any from today, we also have a great email that you can reach out to. So, yeah.
Okay, we do have one question from online, which I'm gonna summarize as, is there a certain threshold of DUX4 that we think we need to hit in order to no longer have the negative effects on the cell? I think that's a question maybe for all DUX4 targeting drug development.
Yeah, I mean, that's a really good question. So, I mean, I'll bring it back to like the mouse model. So one thing that we didn't show in that mouse model is that while with the tamoxifen-induced DUX4 expression, while we did see a dose-dependent decrease in the DUX4 gene signature as we increased the doses, even the lower dose of the mouse version of AOC 1020 still produced a recovery or a similar recovery of the treadmill running and muscle strain. So maybe indirectly answers that, but even at like lower concentrations on the dose-dependent curve, we could still see a recovery of strength and function.
Thank you. We'll have to save the rest of the questions for after. Thank you very much.
Okay, last but not least is Ali, and he's going to give us an update about the first-in-class small molecule inhibitor. Okay, so was it the tools that would lead? Then what was it? It was a view.
Yeah.
Single page? Right?
Actual size or?
Actual size, okay.
Maximize it from there.
And then you can maximize it.
Yeah, okay. So let me share that screen first. Share. Okay. And then here. Okay, that should do it. Yeah.
Right. Thank you so much for having me. It's a huge honor for me to be here amongst world-class scientists and FSHD experts. This is now my second time coming to the gala. My first time was last year, and I remember dancing way more than I thought I would. So we'll see what happens today. Epic Bio talked about targeting DUX4 by silencing the DNA. Miracule talked about targeting DUX4 by targeting the RNA. And I'm going to talk about inhibiting DUX4 by targeting the actual protein. And so by means of introduction, my name is Ali Ozes. I'm the founder and CEO of Altai Therapeutics. We're a small biotech company based out of the San Francisco Bay Area. We're a team of 6 scientists working hard every day, trying to find inhibitors for diseases that have huge unmet medical needs, with our lead program being for FSHD. So Altai is focused on transcription factors, which is a class of proteins that bind DNA in a very specific sequence, read DNA, and then turn on their downstream target genes. And it's a lot of other people have talked about today. Transcription factors are highly regulated. So normally they are either continuously activated or transiently activated, meaning they're only active for a couple of hours and then they're turned off. And then some of them are actually turned off in adulthood. So some transcription factors are expressed only in early embryogenesis or early development, but then are turned off in adult tissues. And so, as you can imagine, since transcription factors can have hundreds of target genes, their misregulation almost always leads to some kind of disease phenotype. Either it's directly related or it causes downstream effects that ultimately lead to disease. DUX4 is a transcription factor implicated in FSHD, as Dr. Tapscott said earlier. So it's turned on in very early embryogenesis and it's turned off after that. And so when it is upregulated either through epigenetic derepression or de novo chromosomal rearrangements, it results in overexpression of the DUX4 protein, which results in its activation of its downstream target genes, which is very toxic to skeletal muscle. And so before I talk about what we do at Altai, I want to sort of take a step back and show you this beautiful structure of this enzyme called HMG-CoA reductase. So this is a really good example of what a highly structured protein looks like. As you can see, it's perfectly symmetrical. If you were to split it in half, it has like mirror image. If you were to split it horizontally, it's a mirror image. And it functions by interacting with a cofactor called NADPH that binds to these very specific binding pockets. And what that does is catalyzes this event that results in increased cholesterol synthesis. So this structure has been defined many times. I think the first time was several decades ago. And what we find now is that we were able to identify where the NADPH binds to, and then we can easily then start designing molecules that would inhibit that site and hopefully turn off cholesterol. And those inhibitors are now called statins. Some of you may be on them, or you may know people who are on them, Crestor and Lipitor. DUX4, on the other hand, looks like this. Very, very different, vastly different protein structure. It has a couple of alpha helices that recognize DNA, but most of the protein is highly, it has this string-like structure, so we call that disorder. So it has these large stretches of intrinsically disordered domains, which makes it very, very difficult to find a nice binding pocket for small molecules. It turns out, however, over 85% of all the proteins in our cells have these large stretches of disordered structures. In fact, only 15% of the proteins that have therapies that are actually drugged, that are FDA approved. There still remain 85% of proteins that could potentially cause disease that remain to be drugged. And so I'm a new entrepreneur. I started Altai back in 2020. And when I read this, I thought, well, 85 is a lot larger than 15. So let's do the undruggables. And so who needs an MBA, right? So it's also presented a very intriguing opportunity, because it also allows us, when we're trying to really take a deep dive into these highly unstructured proteins, you really get to understand the biology of them. And so that's what we've been able to do with the proteins that we've been studying. And so this is an example of one of the targets that we initially started Altai with. It's called STAT3, implicated in fibrosis and inflammation and matrix accumulation, which is actually ironically highly relevant to FSHD. But what we found in our early studies is that STAT3 has these really large stretches of disordered domains that allow it to interact with DNA. But when it interacts with DNA, we're able to capture these transition states where the disorder then becomes ordered. And then when we look at it closer, we're able to identify small molecule binding pockets that can bind and inhibit its activity by blocking its interaction with DNA. We use genetic-based approaches, cell-based assays, and then at the end, we use computational biology and AI to confirm our results. So we did this, so I was presenting this slide to at a mentorship meeting about 2.5 years ago. And someone here in the audience was also there, Greg Block. He was the sat on the scientific advisory board of Friends of FSH, and he said, I think DUX4 is also highly disordered. So do you think you can do this with DUX4? And so I said, I think so, but it'll be a really high risk, high reward kind of project. And we received our very first grant funding from Friends of FSH about 2 years ago to fund this. Because it was highly disordered, our studies with DUX4 resulted in actually far fewer hits than we would usually get from our other proteins. But the silver lining was it actually resulted in one of the most potent hits that we've ever identified. It was only one hit, so we actually identified several binding pockets within DUX4, and we then screened 4 million small molecules against those binding pockets. And of the 4 million, about 200 looked like potential inhibitors. And working with our chemists, we synthesized 40 to 60 of them, and we brought them in our labs and we started testing their activity. So DX5 was one of the most potent hits that we've identified from this assay. Since then, with the additional funding support from Friends of FSH, Solve FSHD, as well as NIH grants, we have now made over 250 analogs of DX5. We have now dozens of compounds that have really, really high DUX4, anti-DUX4 activity in in vitro cells, as well as in the humanized xenograft models. And so we're really, really excited about the future. So this is one example of the humanized xenograft model that was also introduced is with one of our compounds. We have compounds that can repress DUX4 almost 100% in these models. So you can see a dose-dependent inhibition of DUX4 target genes. So this is a composite score of about 8 DUX4 target genes, and we can get dose-dependent inhibition. And last, we also acquired the Flex DUX4 mouse model that was created by Peter Jones and Takako Jones. We've been growing these mice, and basically what the Flex DUX4 mice is an actual functional study. You can induce DUX4 expression by giving mice tamoxifen, which results in DUX4 expression in skeletal muscles, and then these mice really start to lose a lot of muscle mass over time. And it's dose dependent. You can increase tamoxifen, and then you get much faster decline. And so in this study, we treated mice with tamoxifen and with one dose of our inhibitor that we show with the same compound. And then that was 3 times a day for, in this case, 15 days. And we also measured throughout the study grip tests, and then at the end of the study, an Aurora functional test that I'll talk about. So this is examples of what I'm talking about. This is the grip strength test. Basically, although this is a rat, it's a meter. We use this meter, very similar. And you grab the mice from the tails, they hold on to the meter and you pull. And over time, as they lose their muscle function, the grip strength goes down. And over here on the right is our muscle function assay. This is actually photos from our lab, our scientists working on this. So we're able to anesthetize the mice, and we put their foot on a little pedal, and then we can actually put 2 little needles into the tibialis anterior muscle, send electric currents, and we can actually measure contractility of the muscle. So this is one assay looking at just total muscle mouse weight over time. Again, The black line represents no tamoxifen control, and then when mice are given tamoxifen, you see this really fast decline of total muscle weight. And then the green line is tamoxifen with our drug, so it completely rescues even at 30 mg per kg mouse weight. And this is the muscle function with that same compound, and we're able to rescue in a statistically significant manner the muscle function of these mice. So with that, we are very close to nominating a clinical candidate in the next couple of months. And after raising additional funds, then we'll move into CMC and IND enabling studies and go into hopefully phase 1-2 clinical trials at the end of 2025. And obviously, none of this would have been possible with the initial support of Friends of FSH. So even though I love supporting academia, but as a young entrepreneur, as a small startup, I think it's equally important that Friends and other nonprofit organizations continue to fund small biotechs like us. And with that, this is our team of 6, and we also work with world-class experts in drug development, and we're totally an international company. We have people from Denmark, from India, and China that we work with on a daily basis. So with that, I will end there and take any questions.
Couple of questions. I don't think Terry will let everything wait to the auction, so we gotta wrap up relatively quickly. Questions for Holly?
So is the compound, they say you're testing a lot of different compounds to start with, so you narrowed it down to one particular compound?
Yeah, we have 1 and we have 3, 4 backups that are very different as well. Yeah, and it'll be oral, hopefully once a day or twice a day.
Well, maybe you can explain the analog part of it. Is that maybe your question? Like
Was
it's
that-
one, yeah.
Yeah, I mean, these are very small changes within the original hit molecule, but we've also made larger changes, and we're able to, through those changes, we're able to understand not only the structural change within DUX4, but how those small molecules, where they bind and the interactions they make within the disordered regions. So we're not just doing drug development, we're also able to understand really key biological roles within DUX4.
Someone was in right there. Oh.
What would be the projected human dose? I don't think that-
Yeah, so we've done human projections. It's really early to tell right now, but it's anywhere between, we have compounds at 50 mg daily up to 500 mg daily. So it's within that range.
One question online, which I think is helpful to clarify, because a couple of folks have talked about the tamoxifen-inducible mouse. Is there correlation to FSHD patients in breast cancer who get tamoxifen with changes in their DUX4 level expression? So maybe you can explain the difference between an inducible model and an endogenous DUX4 model. Does that make sense?
Yeah, so I don't know if so tamoxifen is the cassette that's introduced in the Flex DUX4 mice is driven by tamoxifen treatment, and so that's what creates the DUX4 allows for DUX4 expression over time. But tamoxifen in the mouse model is usually out of the system within 6 hours, and so we don't think that our compounds are doing anything with the tamoxifen. But in terms of, and we've also tested whether or not, is it blocking tamoxifen, and that's why we're seeing these effects, and we see absolutely no effect in that regard. So I don't think that we anticipate anything with patients that are taking tamoxifen, but I'm not 100% sure.
I think one more question from Rajni. IC50 or KD of DX5 towards DUX4. What was the, yeah.
I mean, the compounds that we have in terms of reporter is in the, it's again, a wide range of as low as 50 nanomolar up to a couple of micromolar. This drug 5057, we see inhibition as low as 45 nanomolar. And so the exposure that we see in these tissues are in the hundreds of nanomolar. So we think that we're able to get at, able to get more drug into the tissues that is higher than the efficacious dose.
Okay, let's think.
Thank you.
Thank you to all our presenters again, and thank you everyone for attending. And please get ready, and we're ready to party in like 40 minutes, I think. So, all right.
Yeah.